The next speaker is Zachary Root. He will be speaking about histological and transcriptional features of programmed genome rearrangement in the C. Lamprey Petromyzon marinus. Take it away if you get your slide. Yeah.
Sweet. Thank you very much. So cancer in humans comes in a lot of shapes and forms, but it's safe to say that this is the product of something in the body going wrong. Our cells normally know what sections of DNA they need to use and at what places and at what times, like a switch. So we can imagine some cancers then as switches that are in the wrong positions at the wrong places or times. Now, a big problem that our cells have is that they don't need to use all of their DNA. The cells in an eyeball don't need to use all of the DNA that a muscle cell does, for instance. Taken in that light, a lot of our DNA is just an accident waiting to happen. But what if we could cut out the sections that we don't need? What if the eyeball cut out the sections of DNA that the muscle cell uses? Now, human cells don't do this, of course, but there are some organisms that do. Our lab, for instance, studies lamprey, and their cells will cut out up to 1/5 of their DNA in a process known as programmed genome rearrangement. The only cells that save it are those that are going to become the testes and ovaries. Now, this distinction matters because these cells, the information they have can act like master switches. If they go wrong, then there's a much higher chance of developing certain types of cancers. If we can understand the mechanisms through which programmed genome rearrangement happens, we could translate this work into humans. So we know when this process happens in lamprey, when they're still pretty young and just a ball of cells like here up in the corner. And the first question would then be, where are our sex cells at this point? How can they be shielded from the effects? So at this point in development, there's only a handful of them amongst the thousands of normal cells there are. So it's a lot like finding a needle in a haystack. Thankfully for us, we can even the odds. We can use state-of-the-art sequencing to figure out what sections of DNA a cell is using. It's transcription. And we can compare the chemical features of these cells and their structural features, their histology. Using these two tools together, we can begin to build a profile of individual cells, compare them to each other, and then compare them to their human equivalents. At this stage, if we can identify where these sex cells are, then we can begin to understand the mechanisms by which they survive programmed genome rearrangement and translate this work into humans as preventative medicine. Thank you.